everybody. I hope everyone enjoyed lunch. Thanks, Steve Emmett Maddox, for organizing that. That was lovely. All right, so I think we're going to get started here. It's a little after 1 o'clock. And as you recall from the agenda, this afternoon, we're going to be digging in on the carbon stocks and database project. So if we can get everybody's attention, Amy Borde is going to kick us off on that. Quick reminder, as we're asking questions and stuff, if you guys can try to remember to grab a microphone, that would be great. And then presenters, if somebody is not grabbing a microphone, if you can try to repeat the question before you answer it, that would be fantastic. There are fewer people here now than there were. Are we waiting? Yeah. Okay, so some people, I see some. Let's give it a couple minutes here before we get rolling. Amy, which one of these is your? Does it say?
I can talk more about the salinity range.
But use the microphone. I don't see. Where's Borde?
Oh, it cut it off. I think it's this one.
Ta-da.
There's some of that monster carrots right there.
Should probably give our team members a little bit of a chance. Like Boone is not here. Chris is not here.
Heidi's not here. Ron's not here.
I'm just going to text.
Judy's
Thank you for being here, Jude.
here.
We were
the only one
taking,
of the team
we
here.
were, and Lisa, we were taking roll about who's tardy. Team members.
Okay, Steve is back, check.
I see Steve Emmett Maddox. Okay. All right, so let's get started. Amy Borde, thank you very much for kicking us off.
Yep. All right, I have the illustrious position of right post lunch here, all I'm sure wide awake, raring to go, barely even here.
We're here for you.
Oh, thank you. Okay, I didn't really look at how I operate this, but okay. So my name is Amy Borde. I'm with the Pacific Northwest National Lab, and I was very happy to be a part of the team that collected the carbon stocks and worked on the database part of this whole project. The folks I listed here were those, kind of directly involved in the data collection and database development, but we want to be sure and acknowledge that the end user group was very much a part of the process along the whole way, where we really valued the input that we got from those folks, you folks out there. So I'm just going to give a very brief overview, and then I'm going to let others talk about what the results were. So I'll just give you a little introduction here. So the objectives that we had were really first to determine the end user needs and the best way for people to be able to access the data that we're producing from this project and other carbon projects. And then also really to fill a big empty niche, which was carbon stock data from these Pacific Northwest wetlands. So we wanted to look at the carbon stocks and the associated environmental variables at 34 sites within Oregon, Washington, and Northern California. And then establish the database to house that data and other data and make it very accessible. And then also to share the results with you all in situations like this workshop. So just very briefly, we've talked a lot about wetlands this morning, Pacific Northwest wetlands. I just wanted to go over really broadly what kinds of systems there are, what we looked at and what we didn't look at. So if you look along, sort of think about the saline gradient there on the y-axis, going from saline to tidal freshwater, and then along the elevation gradient, going from submerged up to forested wetlands. So we focused on the submerged community of eelgrass, really from the saline zone, most saline zone, and then emergent wetlands, both low and high marshes, both from saline all the way up through brackish and tidal freshwater systems. And then we did not look at the shrub-scrub community, which came up earlier today. That is still kind of a data gap that they're very difficult systems to work in if you've ever been in one. But also, kind of hard to find intact systems also, not that many of them. So we decided to really focus just on the ones where we could get the most data. So then we focused also on the Sitka spruce forested swamps. All right, so we ended up with 34 sites throughout the study area. You've seen this map several times now. But the green dots, or green triangles, kind of represent broadly the sites that we worked in. And then over on that side, you can see the breakdown of what the sites were, and then within each of the sites, we had multiple kind of sub-sites within them. So we had 2 different sites, eelgrass sites in Padilla Bay, for example. We tried to cluster the habitat types so that we had multiple different habitats within one estuary. We did that as frequently as possible. So we would be covering the gradient from, at least from marsh up into tidal swamp. And then once we determined our sites, we used a whole lot of different data sources to determine where exactly at the sites we would be doing our coring. We used elevation data and historical land cover data. We also used a Landsat grid, which I don't know if you can see that on the far figure there, to try to be sure that our data where we were collecting our cores could potentially be tied in with data collected through remotely sensed information as well. And then the methods, just very broadly, we, at each site, well, first, there's the breakdown of what the kinds of sites that we ended up, the numbers that we ended up going to. So we had, the most were high marsh, but pretty close with all of the other sites that we had represented throughout the study area. And then at each site, we collected 6 deep soil cores, at least 1 meter deep, up to 3 meters deep in some places. We had 12 above ground biomass samples. We collected data on downed wood along 24 transect lines. We identified all the trees and shrubs within each plot and measured the diameter of those to be able to calculate biomass. And then we also determined the dominant vegetation cover within 12 quadrats. And then for the environmental variables, we also surveyed the site elevations, and we put out groundwater and surface water sensors, which measured water level relative to those elevations, so we get an indication of inundation frequency and the hydrologic regime. We also measured water temperature and salinity. We collected the data over in the summers of 2017 and 2018. So I'm now going to turn it over to my teammates. Boone is going to talk about the results of the carbon stock data. So that's basically all of this data that we collected with the cores and all of the biomass data. And then Chris Janousek is going to talk about the environmental drivers related to the carbon stocks and also some decomposition work that he was able to accomplish also. And then Chris will also talk about the Blue Carbon database and the status of that. So Boone, you're up.
Well, thank you so much. It's a real pleasure to be here today to get to talk about this great study, and thanks, Amy, for starting us off. And I just want to, first of all, acknowledge all the co-authors that are part of this study, and it's very similar to the same folks that Amy just came up with. But Lila Giovanoni, who is my graduate student, who was on all of these sites collecting data with us, as well as undergraduates Jim Kelly, Nick Dunstan, Haida and Amy from PNW, Craig, and all those, and 10 undergrads that helped us throughout the years doing this, resulting in not only the research results that you see today, but a lot of great new blue carbon scientists out there. 2 undergraduate honors theses have also coming out of this study. And of course, you know, we can't do this. It's just like it takes a whole village to do a research project, and that's sort of what this project is. And we've seen this figure of the end users and the project team, and it would not have been possible without all of us working together. And like we said today, the data I'm going to be talking about today, the study areas are the same study areas that you've been seeing all along. But just really quickly, before I get started, I just want you to know that I gave this talk a few weeks ago in Copenhagen. Well, not this talk, but a very similar talk. And I do mangroves. Most of my research in my life has been in mangroves. And for the first time, I was giving my talk, and I was getting real excited about the data, and I fell off the stage. It was like Chris Farley in a Saturday Night Live thing, and I had to get up going like, I'm okay, I'm okay. Nobody remembered a damn thing I said, but they do remember the fact that I gave a talk, you know. So, but I can't, and it was like those things of when you're dying, you know, I'm falling, and I'm going, I can't believe I'm falling off the stage. So I'm so happy that there's not a stage here today, because this is my first time back, you know, on the horse. And at any rate, that's just to let you know that I'm a little nervous because of that. But the reason why we started this project, you know, we have data on blue carbon in many parts of the world, but if you just look at this early study by the commission, the Canadian Commission for Environmental Cooperation, and they did a review of blue carbon in North America and found that particularly in the Pacific Northwest, there was almost no data collected. One obvious little tiny red dot in the Sonoma's estuary at this time. And it was very similar data for seagrass as well. So it certainly, clearly, given the importance of our ecosystems up here, it was worthwhile to study. And of course, just the overall reason why, the rationale, is the amounts of wetlands lost. While Oregon, the overall Oregon has lost 38, Washington 31, of course California 91% of its wetlands have been lost. You know, of course it's much, much higher here on coastal zones as well. So all of this leads us to some of the objectives of our research here. And the first one, similar to what we've talked about, what are the existing ecosystem carbon stocks of these coastal wetlands of the Pacific Northwest, the ones that Amy just outlined, and I'll give you some more details in a moment. How does land use affect these carbon stocks? You know, what are the potential emissions? Looking at the IPCC stock change approach, and what might be the total greenhouse gas losses? What are the relationships of these carbon stocks to environmental variables that Chris will be talking about later, and then I'll talk a little bit about? And can we develop cost-effective approaches to quantification and monitoring of carbon stocks? In other words, if we get a series of of inventory sites, mesh that well with U.S. inventory program, Forest Inventory and Analysis Program. You know, what would be quick and dirty ways to do this? And I think we have the data to do that now. Very similar to what Amy said, just a different graphic. You know, we looked at ecosystems along the intertidal gradient from the basically the intertidal eelgrass communities to low marsh communities into high marsh communities, and transition in this case would be the Sitka spruce tidal swamp communities. Just real quickly, just to give you an idea of some of the sites we sampled and the conditions in which we sampled, here's our Padilla Bay site, one of them with the transect, 100-meter transect, South Slough eelgrass community sampling, of course, at the lowest tidal ranges in the year, so we can get the sediment cores out of the ground. We did low marsh communities. There's tremendous variability in the composition and structure of these sites, as there is in all the marshes. Sarcoconia, this is a Juncus one from the low marsh in Skagit, Delta Island in Skagit. The high marshes, again, pretty wide variation in composition. South Slough, the Carex-Lindbia, Dischampsia, all the way to, the Skagit site being largely dominated by Chinoplectus, the bulrush. And then finally, the tidal forest, largely dominated by a combination of Sitka spruce, but in restoration, let me encourage you, these are very biologically diverse ecosystems. Also, Thuja plicata, the western red cedar, Douglas fir, very many, red alder, a whole variety of species existing in these areas, and very interesting ecosystems indeed. Just another example, here's the Chehalis floodplain. These, again, are perhaps in Oregon. The estimates are, 88% loss of these ecosystems. Just real quickly, just some of our salinity data, and this might give us an idea that we're underestimating the data this morning. These are pore water salinity, and you can see there's a clear salinity gradient here. The highest salinities were for eelgrass, and these are single samples. Well, actually, they're the mean of 6 samples for every site. But they are at the time of sampling, so it'd be during the growing season. And then Chris can give you annual fluxes and how they might change. But you see, as you'd expect, the eelgrass was the highest, 28 to 42 PSUs. Low marsh, 10 to 48. High marsh, 7 to 22. Tidal swamps, 5 to 13, which surprised us. And this was all done with refractometers. And pasture, 7 to 16, similar to the ecosystems they replaced. pH, nothing really exciting here. And one of the things that we also felt is very important is, we're soil scientists as well, is to sample the entire profile. And if you don't go to the bottom of the profile, and I'll talk about the reasons and the rationale for sampling at these depths throughout the talk. But in the cases where soils are deep peats, organics, we had a limit of 300 centimeters. And basically, the above-ground biomass range you see here, this is biomass, not carbon. And of course, the things that are most relevant is the tidal swamps, having 74 to 427 tons of biomass per hectare. Amy talked a little bit about our approach to sampling. And this is a sampling approach that we've used in blue carbon ecosystems from throughout the world. Largely what we do is at the ecotone of the ecosystem, or on the marine ecotone when it's say a low marsh on the edge of the water, we randomly establish the first plot, and then in a direction outward every 20 meters sample 6 more plots. At every plot we take a soil core, again to 3 meters. If it's a tree-dominated one, such as the Sitka spruce, we would measure all the trees, the DBH of all trees within a 10-meter radius. Small trees less than 5 centimeters, and shrubs are only measured in a 2-meter nested plot in the middle. Wood debris, downed wood, would be measured with the planar intersect technique, radiating out from the 4 corners of the site. And then soil core and soil depth would be for soil cores for carbon analysis, and soil depth would be sampled somewhere close to the center of the plots. Very similar, very simple plot arrangement, very simple approach, but it yields a tremendous amount of data. So you do get a lot of bang for your buck from this approach. And also, we would do species composition, of course. Everything's identified to species, both the herbaceous as well as the woody debris, the trees. Just again, the soil sampling, given the fact that it's the majority of the carbon stock, we really have to focus on that. Again, we sampled the entire soil profile. Basically, the soil profile we done here, connote here or define as the depth to an indurated horizon, bedrock, coarse sands, etc. We use a modified soil core developed by us for sampling in wetland soils. Works very well. If anybody ever needs wants one or wants the dimensions of one, contact me, and our machine shop in Corvallis can make them for you. And then we sample, basically we partition the soils by 0 to 15, 15 to 30, 30 to 50, 50 to 1, and 100 to 300. We found this through extensive experimentation. These tend to be pretty relevant and consistent concentrations of soil and bulk density and other properties in these areas and throughout the world. So we have a pretty good global database with this approach now. Just an example, again, this worked quite well at areas even capable of extracting a sample from eelgrass communities. Here's just an example in a high peat area what one of the extracted samples would look like to be taken to the laboratory ground and then run through the analysis. We'll first of all get bulk density because it gives us, we have a known area of which our soil core was to give us bulk density as well as, and then be able to do the laboratory work. And then of course, you know, you can see here is, this is what we used for soil depth is quite unusual. It's an avalanche pole. So buy your get strong avalanche poles, and you can see this one, since our default depth is 3 meters, here's our avalanche pole right here, here's the sampler, and then here's the avalanche pole when it hits the 3 meter mark, and we know that we hit the maximum depth, our default maximum depth we're sampling. And then once we get the soils into the laboratory, we are able to determine then the bulk density, the organic carbon content. We did, just to be careful, we did inorganic and non-inorganic and found that there was very little. In none of our samples did we find any inorganic carbon. So we didn't have to do that second step of separating inorganic from organic carbon. We did organic matter based upon loss on ignition, and I'll talk about why that's important. And we've also done a second study of looking at loss on ignition versus hydrogen peroxide digest. Loss on ignition is much more efficient in calculating your organic matter. And then finally, we're doing soil texture to get an idea of how can we correlate soil texture to carbon stocks, because I think a lot of the variations, it's one of the most, again, one of the great unknowns and that clearly we know influences soil sequestration and you know, holding in the soils, yet there's been very few studies in blue carbon ecosystems of soil texture. And again, just finally, just measuring the above ground biomass, you know, by clipping plots, identifying the species you see in the upper left beforehand to get an idea of the composition and structure, and of course for the Sitka spruce, we do it with both the trees and shrubs and grasses. Just real quickly to give you an idea of some of the data, this is non-metric multi-dimensional scaling results on the first axis, and it really is cool for just it kind of gives you an idea of when we talk about low marsh and high marsh, we're really kind of setting an arbitrary gradient of a true gradient that exists along these areas of the world. And here's just the abundance of the species along this multi-dimensional scaling axis, which is largely correlated with increases in salinity and decreases in bulk density. And one can see just these wonderful ways that, you know, at where Carex lindbii maxes out right here in the middle, or actually this is a Gros's stolonifera maxes out like in the middle of the salinity areas. And of course, you see sarcocomia and Sarcomium gesticulus, highest in the saline areas, and so you can see this great how all these different species relate to one another. Thanks. Sorry. Yes, but I'm a biologist and I can't figure out how to use this. So. I'll turn off the damn thing if I touch that. So, okay. So, sorry. Okay, so let's just look real quickly at all 34 of our sites. And here's what the biomass looks like. And these are all, and sorry that my legend didn't come up today, but the dark, these dark olive ones are the eelgrass communities, and the light greens you see are the low marsh, the blues are the high marsh communities, and then the dark greens here, the tall bars, and this is total ecosystem. Carbon stocks are the Sitka spruce, and as you can well imagine, the highest ecosystem carbon stocks are the Sitka spruce sites, and you just see a gradient that about what you'd expect to see in total ecosystem carbon stocks. If I just separate these out now by the eelgrass communities and the high marsh and the low marshes and the tidal swamps, you see a different story starting to emerge, where as you would well imagine, the eelgrass are the lowest, the Sitka spruce are the highest, and then just taking the means of all of these sites now. And this, I think, is the cleanest data set that I've ever sampled in my entire career, in that we see that everything is significantly different in terms of ecosystem carbon stock. So that's one way to start to partition the ecosystem and simplify and improve your accuracy in the future will be to, you can use this now as tier 2 default values, but you see that the Sitka spruce swamp is almost 1,000 tons of carbon per hectare. The high marsh a little bit higher, about 600 on bean, 400 for the low marsh, and about 225 for the eelgrass communities. You see that basically the 1 meter, the soils, almost half of it will be the soil, the deeper soils, the soils 1 to 3 meters in depth, or soils greater than 1 meter in depth. And again, probably about another third is the soils in that top meter of soil as well, and above ground mass being very significant in the Sitka spruce. Yeah. That's just the way they're arranged on this. Yeah, I kind of arranged them like that on purpose, just to go from low to high. Okay, but anyway, the thing that's just so incredible, though, is that soils are significantly different between the 4 different types. Above ground biomass, obviously significantly different, and total ecosystem carbon stock, significantly different. If we just look very quickly at the eelgrass communities, we see the mean of the eelgrass communities is, again, about 225, but there's tremendous variation in ecosystem carbon stocks. In the lower Padilla Bay, one as small as less than about 48, 47 tons per hectare. And then Humboldt eelgrass and Sally's Bend eelgrass in Yaquina Bay, very, very high. Some of the highest seagrass community, ecosystem stocks of seagrass communities ever measured were these 2 sites. And the reason being, I think, is these are finer textured riverine sediments tending to sequester more carbon in these areas, and we'll have better data with our soil texture analysis later on. If we look at the low and high marshes, again, we see, the similar huge range in total ecosystem carbon stocks, anywhere from 175 at Delta Island to as high as over 600 at White River Marsh and Humboldt Bay. So again, tremendous range. And then just looking real quickly, let's do a quick comparison. We do have data using very similar methods from other marshes of the world, and you can see that here's the PNW low marsh, a little over 400 on average, the PNW high marsh at a little over 500 on average, and then marshes in Mexico, in Brazil, and then the IPCC default value for marshes is, somewhere around 250. So you can see that on average, the marshes of the Pacific Northwest are much, much greater. Yeah. The depth, the soil depth are the same for all of these. They're not for the IPCC. The IPCC default value would vary quite a bit. They'll have an average for above ground and an average for soil. But all of these were measured by me, so they are the same, largely the same methodology. So. Oh, and what's really cool about this, though, is this is the tall marsh in Brazilian Amazon, 0 degrees, and it's Chinoplectus. And here's the low marsh, ecotonal to mangroves, and it's Distichus spicata. So it's amazing. It's the same species, even. Okay, so if we look at the swamp data, we get a whole great story here. We can see this is the total above ground ecosystem carbon stock. So this is just the carbon stock above ground. We see it ranging from quite low here at Otter Island, about 75 tons per hectare, to the highest area, the Secret River Swamp on the Columbia, at about 450 tons per hectare. You see that the green is the tree carbon. So this is going to be difficult in restoration. It's not just planting trees. To get back the ecosystem structure, almost half of the mass then is wood that's sound, wood that's rotten. This is the herbaceous carbon mass, and this is root mass right here based upon a regression equation as well. So again, it may take centuries to truly restore an old growth forest. But what you do see is, notice the high wood content here at Secret River Swamp, at Blind Slough, and at the Chehalis Surge Plains. Those are truly undisturbed old growth forests. I think that Coal Creek, Johns River Swamp, Otter Island were secondary forests. We know this because there's stumps out there. So these are really secondary forests. But one of the things it shows you is maybe they were pasture 100 years ago, 40 years ago. We need the aerial photos or remote sensing data. But and we get the idea largely from that lesser amount of large wood in the ecosystem, even though they've grown back quite well. You see tremendous amounts of tree carbon in these areas. So again, important ecosystem carbon stocks, especially when they're old growth forest or undisturbed forest or late successional forest. If we look at the total ecosystem carbon stocks of these tidal forests, you can see here again that the majority of the above ground biomass, the majority of ecosystem carbon stock is not the above ground biomass, but again, the soils. Here's the 0 to 15, 15 to 30, 30 to 50, 50 to 1, 50 to 1, and greater than 100. That's roots right there, sorry. And again, but what really surprised us, I think, was the fact that the consistency of these things. There's hardly any variation in the total above ecosystem carbon stocks of these sites, but they're consistently extremely high, well over 1,000 tons of carbon per hectare. So these are very, very large carbon stocks. Now, you might ask how large? Well, let's look at some comparisons here. And we can look at forest inventory analysis data from and old growth forest data from the literature, upland forest. And this is based on forest inventory analysis of all forests, regardless of their age. The average in the Northwest is about 275 tons of carbon per hectare. These are just, if we look at coast range forest, which are the big forest in the Pacific Northwest, it comes up to about 375. And then if you just look at literature, data, particularly from Smithwick et al., where she looked at the ecosystem carbon stocks of old growth forest, and they're about at 700, almost 700 tons per hectare. If we take our mean of marshes and swamps together, it came out to about 350, and that's down to 1 meter. And the reason why I just did this comparison, because this is based on 1 meter as well. But I would suggest that the real example, what we need to compare, and I'll talk about this in a minute, is the total horizontal carbon, and that would be this side here. And this, again, is marshes and the swamps together, combined, just the mean blue carbon of the intertidal region. And of course, remember, it'd be 1,000 if it was the Sitka spruce forest alone. But the reason why this is pretty relevant, the sampling the soils in mineral soils and uplands to a meter, because that's about the zone of influence. But what we have found throughout blue carbon ecosystems, and blue carbon ecosystems are disturbed. You disturb, and the sources of greenhouse gas emissions are far greater than 1 meter. And so it's very, very important. So I would suggest that this figure of down to 3 meters is relevant as this is carbon that's vulnerable to mineralization, oxidation, and loss with land use, land cover change. So another quick example or comparison is how do these compare to other blue carbon ecosystems? And here's our mean tidal swamp again, the low marsh, the high marsh, the eelgrass. And here's just an average of mangroves throughout the world. Average for mangroves based on 190 sites is was about 825 tons of carbon per hectare. And you can see our tidal swamp mean is about 1,000 tons per hectare. So this makes these sites about the biggest blue carbon ecosystems on the planet, really. There are some mangroves that are this big as well, but the mean is pretty high. So these are very important ecosystems. Not only are they unique, but they also are capable of sequestering a large amount of carbon. Okay, so that's it for carbon stocks, but let's go to the second question, and that is, how does land use, land cover change affect carbon stocks? And now this is what's really important, and we need to talk about this. How do you compare a pasture to an adjacent wetland? And this is the Hepel Marsh, and basically it's really important to get paired areas where you can make the assumption that the pasture was the same as the marsh or the Sitka spruce forest before it was disturbed. And it really requires that you need adjacent sites if you can possibly accomplish this. Now, when you compare the carbon stocks, it would be real easy if we just could take a core and then compare the mass of one site to the next, but you can't do that. It'd be the reason why it'd be an analogy kind of like this. Say you get 2 loaves of bread, and on your way home from the grocery store, you sit on one by accident, okay? And you come home and you pull out 3, and that bread is all smashed, the one where you sat on it, you know? And now you pull out 3 or 4 pieces of bread, and it's the same thickness as 1 piece of bread. And you go, well, there's more wheat in this little sat on bread, so it must be better bread, right? Well, that's exactly what you're seeing here, is years of subsidence, trampling damage, loss of root structure. You have higher bulk density here. So you can't really sample. So when you're sampling, if we sampled the 2 meters, we'd be sampling you're sampling the same volume, but you're sampling a different mass. So what we do is what we call the mass equivalence measure, where we need to then calculate the mineral mass of the top meter of soil here in the wetland and then compare it to the depth, or excuse me, we did the opposite. We take a meter of the soil in the pasture, do the organic matter, separate organic matter from mineral soil, take that mineral mass to 1 meter, and then do an equivalent amount of mineral mass in the wetland to get an idea of how you make that comparison. So, in other words, we're comparing on the basis of mass of soil, not a volume of soil, which I think is much, much more relevant given the changes in soil properties. Okay, if we do that then, and ours were really not very subsided ecosystems. On this side, it was 1 meter of pasture was equivalent to about 168 centimeters of soil in the wetland or the marsh, and here it was 1.3, and here at Millicoma it was 1.86. So what we see then is that the overall mass loss of this site was about 175. Here it was quite remarkable, almost 400 tons of carbon per hectare, and here again another 200 or 300 tons per hectare. If we convert this then to CO2 emissions, you can see that the CO2 equivalent loss at Hepel was 489 tons per hectare. Johns was 998 tons. This is just the molecular ratio you convert by multiplying the carbon by 3.67. And then the South Brothers was 735. But again, the overall average of these three sites in was 741 tons of CO2 equivalent loss per hectare in these pasture areas. So it's pretty significant loss in carbon. If we look at, this is just, again, some global data of emissions. Here, the blue and gold, I'm not a Berkeley fan, but it's just, these are the blue carbon ecosystems, and these are terrestrial forest ecosystems right here. And the little star here is our average loss of the pastures here in the Northwest. So it's really quite low compared to the other blue carbon ecosystems. These are all either conversion of mangroves to pasture or mostly conversions of mangroves to shrimp ponds. And there we lose all of that above ground biomass, huge carbon stocks in the forest, as well as pretty significant losses in the soils. But the thing that's interesting is it does exceed the emissions lost in the Pacific Northwest, does exceed, say, the conversion of an Amazon rainforest, which is about 710 versus 760. So it's pretty significant loss. And this is just the loss of pasture as we get studies down the road when we start to look at the loss from the Sitka spruce forest. They're going to be probably equivalent to some of these mangrove emission losses. Okay, one last thing that I'd like to talk about then is not only are we important in carbon markets, but coming back to what we've been seeing in the news the climate strike last week, and Greta Thunberg and others, is not only is there a market value, but there's a social cost of wetlands loss. And the social cost of carbon is something that was largely developed during the Obama administration and the Clean Power Initiative that looked at the economic cost caused by an additional ton of carbon dioxide emissions or its equivalent. How do future generations, how much are they going to have to pay for emissions today? Early on in the Clean Power Initiative, it was basically $31 to $36 per ton. Today, it would be about $42 per ton. Nordhaus and EPA are the sites I'm exampling. But if I just use Nordhaus's data at $31 per ton, the emissions from marsh conversion to pasture give us a social cost of about $15,000 to $36,000 per hectare. So the social cost of carbon is quite high. And in fact, if I was even trying to sell participation in voluntary carbon markets, I'd say, but you're paying $10 a ton, but if you look at the social cost of the carbon that you're sequestering, it's much, much higher. So it's a really good, I think it's a selling point that we ought to be trying to push people for voluntary carbon markets. And it's also important to remember that this doesn't include, as we talked earlier, greenhouse gas emissions from land use, the methane and N2O emissions that may come from, say, livestock or from agricultural operations. And the conservation, restoration has economic values, not only for the carbon, but environmental, health, cultural, and biodiversity values. Okay. So just in conclusion then, that it's really apparent to us that blue carbon ecosystems, Pacific Northwest, significant sinks and stocks of carbon. There's tremendous variation in these carbon stocks, suggesting that we need to know the carbon stock of your site in question. It'd be hard to use a single site. Eelgrass, all of them have tremendous variation. There's been hypotheses and suggestions that there's such low variation in total ecosystem carbon stocks that you don't even really need to measure it. That's clearly not supported in this study. Even if you limit the sampling to 1 meter, it's still, threefold differences in eelgrass, twofold in high marsh, twofold in low marsh, and then, actually only about a twofold if you include the above ground biomass in the tidal swamps as well. But if you conclude the entire soil horizon, it's even much greater variability. This is your own conclusion. No, sorry. Land use results in significant emissions of carbon, and this is social carbon cost of $15,000 to $31,000 per hectare. And it also suggests that conservation and restoration would sequester significant quantities of carbon. And we see this. One thing about these ecosystems is they are pretty resilient. They do come back quite rapidly. You know, in some of our areas, some of our just preliminary data, we're seeing up to 8 tons of CO2 equivalent sequestered per hectare the first 10 years with rest. So there's more data needed, but there is pretty suggestion that the resilience and that the recovery of these is possible. And we need to be a mechanism to be part of state national carbon inventories. How do we meld? I think we need to have these areas starting to be considered, collected, and combined with the forest inventory data so they can be part of the problem or part of the solution and not ignored as they've been in the past throughout the world. And with that, what we have left to do still is we're still analyzing. We've done all the work. We're analyzing soil organic matter, soil texture influences on carbon density, so we can have cost-effective approaches to soil carbon stocks. That if you know soil texture, bulk density, soil organic matter, really things that are pretty easy to measure, you could get a pretty accurate estimate of your carbon stock, and this would help us have region-wide carbon inventories of blue carbon. And emissions from the conversion of the peat swamp forest is ongoing research as well. And how do we incorporate this into existing carbon inventories? With that, thanks a million. So, any questions or comments? Yes. Yeah. Okay.
Okay. I'll keep it down to one at this point. So my question, though, has to do with horizonation. Have you actually looked at any of these soils based off of, like, the deposition? Because one of my experiences in looking at the Quileute soils was that it was very patchy. So, like, you would have, like, sands, and then you would have some silts, and then you'd have, like, this thick horizon of sheer organic matter with, like, visible spruce cones.
Mm-hmm. Mm-hmm.
So to me, horizonation was like a critical part of understanding the soils that I was looking at.
Right. Well, you know, the reason why we don't do horizonation is because we're trying to do this throughout the world, and every site has a different soil horizon. You know, what we have found that the 0 to 15 typically does hit the A1 horizon. You know, 15 to 30 typically hits the A2 or the B1. And also, remember, we're taking a 5 centimeter depth core, pulverizing that, grinding it up, and we do this, we have 6 replicates. So that amount of replication tends to cover all of the variation that you had. Now, we do see, on rare, you do see, you know, here in particular, we'd see, you know, you'll see these little layers where forest fires came through, and you'd see the little ash layer through. Yeah, we don't hit that. We don't hit that much. So there is some loss, but you have to sacrifice that degree of precision to the ability to get the work done, to do the project. But, you know, what we've done, of course, we've done this, we've sampled every centimeter, we've sampled every 10 centimeters. You know, we find that these are pretty uniform soils by and large, particularly when you have a sample. Our minimum sample size through statistical analysis was 6, and therefore we're pretty confident at the accuracy of our data here. And we could
So our
talk.
follow-up question. So you homogenize each of those segments. So when you're taking the 1 to 300, that's a much larger
No,
amount of soil?
we sampled, the 0 to 15 is sampled independently at the 15 to 30.
Yeah.
Yeah.
But then, so like,
And all 6 are sampled, all
so
6
like the
sites.
50 to 100, you're doing 50 centimeters of homogenized soil?
No, just we'll take the middle 5 centimeter.
Okay, so you would miss any of the micro?
Yeah, right,
Okay, thank you.
yeah. Unless, you know, we would miss it if it happened to fall, if it falls right in the area where we're sampling, we would miss it, we'd be sampling it.
Okay, so I'm not a soil scientist. When you say soil texture, can you say more about that? How do you determine the texture of the
That's
soil?
the percent sand, silt, and clay in the soils. And what we have found is that the higher, that there's a direct correlation, we haven't found it, many other people have found it, there's a direct correlation between carbon density, the amount of carbon per square centimeter of soil, and the finer textured soils. So what we would like to find out is how well does that hold up throughout these varying environmental variables. But the hard thing is, in wetlands, it's very difficult. When you're over 2% organic matter, you need to digest the organic matter, so it's an extra, it's not easy to get sand, silt, and clay, but first you have to digest the samples as well to get rid of the organic matter. So that's a second step that's unique to wetlands. So hopefully we can do it so you won't have to later on. So yeah. Thanks.
And how we compare here with the other parts of the world is really good. But
Yeah.
I keep thinking about the eelgrass, and the thing about the above ground turns over maybe 7 times a year. So the turnover rate's pretty high, and that stuff goes somewhere.
Yeah.
And where does it end up, and what happens to that? I think it's just like I mentioned earlier. I think it's an important issue to look at.
Yeah, it's one that has been in Copenhagen a few weeks ago. It's the Blue Carbon Working Group. It's something that's being debated ad infinitum between the seaweed people as well as the seagrass people, and where does it go? Where does it end up? How much is mineralized? How much makes it to the long-term storage? It's a great question.
I'd like to hear more about it sometime.
Yeah.
I'm not familiar with what's happening in Copenhagen, but
Yeah.
I mean, clearly those seagrasses and kelp and other algae, if we're going that direction with carbon, blue carbon sequestration, then it's going to depend on where you are on the coastline. If you're doing that restoration on the outer coast where it's going to get sloughed off into the deep sea, then that above ground vegetation could be counted, and not just the below ground, the roots, et cetera, that's getting down into the soil. But if you're in the Puget Sound, that gets a little more complicated, or shallower parts of the Salish Sea. I mean, they're deeper parts, but it's complicated. Well, parts of it, right? So I think it depends on where you are and what how the currents are pulling those different vegetation out. I don't know if anybody's looking at that.
Yeah. Okay. Any other questions? Yeah.
This might be covered later. I'm interested in kind of if there's any data or plans for data on shorter timescales for like these carbon stock changes.
Yeah.
I don't know if you can speak to that, or maybe Chris is going to speak.
Yeah,
to that.
I think the next phase would be, it would be really great if we did get into, well, there's 2 ways that we'd approach restoration. We need to look at trace gas sampling and getting that to ecosystem carbon balance. In other words, how much carbon's coming in, how much carbon's lost. So, both field measurements of carbon stocks, gross primary productivity coupled with trace gas emissions on an annual basis to get an annual net carbon sequestration, coupled with, say, maybe the rates of sediment deposition. But the other ways too is we could also begin to look at, first and foremost, a crude way would be, stock changes of how much carbon has come in. If we know we have some wet meadows that are restored marshes that are 10 years old, 15 years old, 20 years old, how much recovery have we seen there? If we can find a relevant base mark or baseline to measure? Because what we do see is we've done this in freshwater wetlands, and it's dramatic, the difference. The bulk density recovers in 10 years, water holding capacity recovers in 10 years, infiltration, lateral movement of water increases, and we get an average of about 8 tons of carbon per hectare, a CO2 equivalent per hectare per year in that first 10 years of these very productive freshwater wetlands. So it's something that we need to look at for sure. All right, thanks again.
Okay, so I'm Chris Janousek with Oregon State, and I'm going to build off what Boone has presented in terms of the carbon stocks and how those stocks at all our various sites may relate to the ecosystem drivers that we measured as well. So our major research questions were to try to understand how blue carbon stocks, all this variability that Boone has described, are related to major environmental drivers from climate change, or climate indices to site level data on salinity and inundation, and the elevation within our wetlands within the tidal frame. A side question that sort of came up as we were doing this project, I've been interested in organic matter decomposition for a few years. I thought this was an amazing network to test questions about decomposition. We had all these other data, and so I threw in some litter bags as well to see how decomposition may be related to stocks and environmental drivers. And then the kind of a more applied question that comes from this work is, can we use environmental driver relationships between with stocks to sort of inform our wetland management and restoration? So there are a variety of processes that could affect stocks. So sort of what we end up with after hundreds or thousands of years of carbon accumulation is really a function of a whole bunch of coastal processes, including plant production, decomposition, export of material out of the wetland, and then import through allochthonous inputs. And those processes themselves are affected by a variety of abiotic and biotic drivers. So this is probably an oversimplified diagram here, but I've illustrated a couple of the factors that might affect all of these processes, including precipitation patterns, temperature, salinity, inundation, irradiance, your tide range, your geomorphic setting, and then a grain size, like Boone mentioned. So these are all interacting factors, and we've looked at a couple of these in this particular project. So these are some of the hypotheses that we'll try to talk about today. The first was that sort of if you look at the Pacific Northwest as a region, we have variability in climate, not a lot, but we have some variability in climate. Is that related to carbon stock values at our sites? And our working idea was that carbon stocks would increase with precipitation, largely due to lower salinity and higher productivity, but might decrease with temperature because of higher decomposition rates. The second hypothesis was focused a little bit less on the sort of region as a whole and more on sort of the site scale. And the questions here are whether carbon stocks are higher in less saline wetlands, as we might infer from the literature or our understanding of how salinity affects plant productivity, and whether carbon stocks are greater where there's increased soil saturation with groundwater, so you have an anoxic environment that better preserves carbon over the long term. Then the final hypothesis was sort of, we drilled down in spatial scale even a little bit more to ask whether carbon stocks are positively correlated with tidal elevation. Some research we've done on elevation effects on productivity and elevation effects on decomposition suggests that that sort of balance between carbon gain and carbon loss might differ a little bit with elevation. So we wanted to test that out with this data set. So you've seen versions of this map, a different version here, but we had 34 sites and 9 estuaries from northern Washington to Humboldt Bay in California. And we collected environmental data from all of these 5 habitat types, wetland types that Boone talked about. Some of the data are only from a select subset of these ecosystem types. And these are the data sources for the ecosystem driver data that are related to the 3 hypotheses that I'll talk about. So for the climate variables, I used the PRISM model. So it's a model developed at OSU that takes 30 plus years of climate data on precipitation and temperature and gives you climate normals, sort of average conditions over several decades. And that data set is available sort of at the site level for our locations. The National Renewable Energy Laboratory, I never thought I would be downloading data from the Department of Energy before, but they have a nice irradiance model. Sorry. they have a nice irradiance model that actually predicts how much incident light reaches different habitats, and that's, of course, a function of fog and clouds and latitude and so forth. So that gave information on mean irradiance for our sites. For the second set of hypotheses, we went into the field and we deployed loggers to measure salinity and the groundwater table. So we have nice time series of both salinity and groundwater level. And we did this in the channels as well, but I won't talk about the channel data. And then we used litter bags deployed for about a half year at a subset of our sites to look at actual rates of organic matter loss over time. And then lastly, to measure wetland elevation, we used RTK, GPS, and in a few instances leveling from known elevation points in order to assess the elevation of all the cores that were collected in the project. And then also the VData model to give us values on the local tide range, because we have variability in tide range within the region and within sites within an estuary as well. So I'll first talk about the larger scale climate drivers. Again, the data sources were the OSU PRISM model and the irradiance model. The climate data come from a 3-decade period, and the irradiance data are available for about an 11-year period. We used GIS to essentially overlay our sampling points with these model grids and extract values for long-term precipitation, winter, summer temperature, and then irradiance. We did this for all habitats, but the analyses I'll show are just on the marshes and the tidal swamps, just those habitats in the upper tide frame. The metrics extracted were annual mean precipitation. We got monthly temperature values, but I'll just show the summer data, and annual mean irradiance. So this is what the gradient in precipitation looks like for the Northwest. It surprised me. I live in California and although work for OSU, and I'm used to much less rain than we get up here, so it surprised me that we had such a gradient in precipitation across the region. So we have areas such as Puget Sound, which is sort of behind the Olympic range, rain shadow, and gets less precipitation, and then we have very wet areas such as the northwest corner of Oregon. So we had more variability than I thought across the region. These are the results that look at a couple of different carbon metrics and how they relate to these precipitation gradients. So none of these are data from low marsh, high marsh, and tidal swamp sites that Boone summarized, and none of the variables that we looked at, carbon content, density, carbon to nitrogen ratio, and then the sort of stocks in the upper soil profile had any significant relationship with precipitation. The next variable we looked at was summer temperatures. So summer temperatures do vary somewhat within this region. Winter temperatures vary as well, and actually sort of the range, the difference between the annual summer and winter temperatures also vary across the region. And sort of similar to the precipitation story, kind of contrary to the working hypothesis, none of the carbon metrics that we looked at had any relationship really with summer temperatures. So irradiance did not vary a lot. So we had some irradiance differences within the region, but by and large, rather similar amounts of incoming solar irradiance across the region. And similar to the precipitation and temperature story, did not find any relationship at the site scale. So to summarize the climate data, there really were there's no evidence for any strong relationships at the regional scale. Part of that might be due to the fact that there is sort of some homogeneity within the Pacific Northwest ecoregion. We don't necessarily have a huge range in temperatures or irradiance. And there were some evidence, I haven't done the statistics yet, but there may be an effect, climate effect on the maximum carbon content and stocks that you would get in your ecosystem. And this is something we can look at with quantile regression, probably with a larger data set. So even though this data collection that Boone led is sort of unprecedented in its size. There's a lot of noise in these relationships, and probably many more sites would be needed to do this sort of analysis. So I'll talk about this a little bit more in the next database talk, where we have even more sites and we have a broader geographic range that includes wider variability in temperature and other climate variables. Okay, so the second question was how do some of the site level drivers, such as salinity, inundation, and decomposition, potentially relate to carbon stocks? To measure groundwater salinity, we deployed Odyssey conductivity sensors for at least a year, in some cases a year and a half. These were placed in groundwater wells that were sunk into the ground about 1 meter down. The sensor itself was usually positioned about 25 centimeters below the soil surface, so that it captured salinity conditions within the root zone, more or less. And we processed the data to estimate mean, minimum, and maximum salinities over the time series. And we just deployed these groundwater wells only in high marsh swamps and a few of the pastures. We didn't have the manpower funds to put these everywhere, so we did a subset of sites. We're still working on the data processing for these, but I'll show a couple of results for the tidal swamps. We measured groundwater table with pressure transducers inside the same groundwater wells. These were positioned right at the bottom of the well, so about 1 meter below the surface, and they captured all of the groundwater fluctuation over that same time series. We also, at our sites, measured barometric pressure so that we could compensate for that effect on water level variability. And again, the same sites measured high marsh swamps and a subset of the pastures. And we are still working on these data sets as well. These time series are very data intensive, but we'll be calculating groundwater amplitude and the duration of saturation within the root zone. To measure decomposition, we placed litter bags with organic matter out into the field. And I came across this paper, Janousek et al., a couple of years ago, and thought it was sort of an ingenious method. I've done litter decomposition with a variety of substrates, usually native plant species that you grind up and put in a litter bag. But they published a very detailed analysis of how the exact decay dynamics of these two types of tea. So you can get Lipton tea. You have to buy it from Europe because you want to buy the exact same type that they use in their study. But you get green tea and rooibos tea. You place these in the soil, and they've really worked out the decay dynamics and the exact biochemical composition of the tea and so forth. So the rooibos tea decays very slowly, so it sort of sticks around for a long time. And then the green tea decays very, very quickly. And they give you sort of different metrics. So the rooibos tea gives you sort of an initial decomposition rate, and the green tea gives you an estimate of how much refractory carbon you have. So how much carbon is not really subject to short or intermediate term decomposition. So the standard method is to, they just take the tea bags and put them straight in the ground, but that can cause problems with roots and stuff. So we put them in these sealed Versapor litter bags, buried these in the ground, again at a select sites where we had groundwater wells, so we could look at the relationship between groundwater and decomposition as well as the carbon stocks. And then we calculated the loss rate K, as well as what Janousek et al. Calls a stabilization factor from the green tea, and that's just sort of an estimate of how reducing and how prone to organic matter preservation your environment / microbial environment is. So they give that a term called the stabilization factor. So the higher that is, the more potential you have for longer term carbon sequestration. And they've published data using the tea bag method for a variety of ecosystems, from deserts to forests. And there's a very large collaborative project led out of Australia that's using these tea bags to measure decomposition rates in tidal wetlands such as eelgrass and salt marsh throughout the world. So we use slightly different methodology and probably can't contribute to that, but there's a cool citizen science effort to really understand decomposition on a global scale with these simple tea bags. So these are some data here that I have from a separate project in San Francisco Bay. This is the percent mass loss over time, and we've had these litter bags out. This is not the tea per se. We actually used native species litter in these, but we have litter bags out for 3 years. We have a few more in the ground that we'll probably collect after 5 years. But these are sort of the typical decay dynamics that you would see with litter. And the rooibos, or the red tea, that gives you that initial curve on the left side, that initial rate of decomposition. And then the green tea, as I mentioned, it gives you sort of this long-term stabilization. All right, so these are some of the results from the tea bags that were placed out in the field. So we have high marsh, pasture, and tidal swamp where we put the tea bags out. We typically had about 5 or 6 sites per habitat class. And we found some differences in both decomposition rates and stabilization factor among the wetland classes. So the pastures, interestingly, had higher rates of decomposition. Litter was decaying quite quickly, but they also sort of counterintuitively also had a higher stabilization factor. So it looks like the organic matter that is in the pastures, which probably have more oxygenation of the sediments, it was decaying quite rapidly, but there was something about the pasture environment that was stabilizing whatever organic matter remained. So the labile stuff went away quickly, and then the rest seemed to stabilize. But these rates suggest that perhaps anywhere from 10 to 20% of, if you're sequestering tea for the long term, 10 to 20% of that carbon has the potential for long-term sequestration. So a lot of it really is potentially decomposed over time. We wanted to see how the decomposition rates would relate to some of the carbon metrics. So these are plots of decomposition rate. On the top, the K value, so that would come from the red tea, and then the stabilization factor from the green tea on the bottom. And we looked at both carbon content and carbon density. And sort of similar to the climate story, we didn't find a lot of strong relationships, but these are also a variety of habitats mixed together. And if we had larger data sets from a single habitat, we potentially would see some relationships. Okay, so these are some of the salinity data. The data sets are quite large, and we're still working through them. But these are some groundwater salinity data from the tidal swamps. And I have part of the time series of 5 tidal swamps here, color-coded in the top and bottom panel. So this is the June to end of August, that 3-month summer period for Snohomish here, Nehalem in northern Oregon, Chehalis in southern Washington, and then our 2 sites on the Columbia River. So interestingly, we had, well, not surprisingly, we had an increase in salinity as the summer season progressed. The peak salinities in our marshes and our tidal swamps are probably in the late summer and early fall before the onset of the next rainy season. Snohomish and Nehalem tended to be our most saline sites in this study. This is the maximum daily groundwater value. Chehalis may have a little bit of salt from time to time, but the 2 sites on the Columbia River, Secret River and Blind Slough, were actually quite fresh. And then I've just plotted the, not the full soil stocks for the 3 meters that Boone sampled, but just the top meter of soil stocks. And there does seem to be potentially some correlation, some negative relationship with salinity within the tidal swamp. So our 2 saltiest sites, Snohomish and Nehalem, had somewhat lower carbon stocks, and then our particularly Blind Slough had very high carbon stocks. So these are just soil stocks. They don't include the total ecosystem stocks that Boone calculated with the above ground biomass. Okay, and so the final area to talk about are the within site elevation gradients. You guys been out in the field know that marshes and tidal swamps have a lot of elevation heterogeneity, and that correlates very strongly with their inundation and soil redox and all these properties. So this was measured for the most part with RTK GPS, which typically gives you anywhere from a 2 to 5 centimeter level accuracy in the vertical. In a few instances, leveling was used from a known point. RTK GPS gives you a geodetic elevation, but organisms don't care about geodetic elevation. They care about inundation and tide range. So we needed to convert that to a tide range scaled elevation. The value I use for that is Z star, and I'll show you that in a minute. So we collected elevation data on all of our wetland types, eelgrass, marsh, swamps, and pasture, and then used the NOAA VData model to convert that to a tidal range. So on the left is just sort of a schematic of what our tides look like in the Northeast Pacific, where we have 2 highs and 2 lows each day. Mean high water is often used as sort of a boundary between high marsh and low marsh. And most of our vegetated tidal wetlands occur, seagrasses being the exception, but the swamps and the marshes tend to occur between about mean tide level and the extreme high water mark. On the right is a figure of, these are just NOAA data from tide stations within the Northeast Pacific, from Southern California up to Alaska. You can see that as you move farther north, your tide range increases quite a bit. The dotted box shows all of our Pacific Northwest region from Northern California up to Washington. So because of that range in tidal variability, you can't use a geodetic elevation from GPS. You need to sort of somehow account for your variability in tide range. And the way to do that is to calculate Z star. So Z is your measured elevation in the field, and then you relate that to the tide range by computing local mean high water and local mean tide level. Okay, so the first couple of figures are just some of the elevation differences that we had among our sites. So these are the 6 seagrass meadows that we had in the study. These box plots show 6 cores, so we took wherever possible an elevation measurement right where Boone's team sampled for sediment stocks. And most of our seagrass sites are around mean low or low water, so they're exposed some of the time, but submerged most of the time. But there was some variability in elevation among the sites. These are the tidal swamps. These cluster mostly around local mean high water. 3 of the sites are a fair bit above mean high water. So these are areas in the marsh that are probably flooding, or excuse me, in the tide frame, that are probably flooding anywhere from 5 to 10% of the time. Perhaps even a little less, perhaps like 3% of the time being above mean high water. Our marsh sites were actually a lot more variable than I think we planned. When we were doing the planning for our sampling, we, as Amy mentioned, we use GIS in a variety of layers. We were LiDAR, for example, and we were trying to capture sites that were sort of above mean high water as our high marsh sites, and some a little bit below mean high water as our low marsh sites. But we found actually, when you go out in the field and collect the data, that we had quite a bit of variability from our sites. In particular, our 2 Columbia River sites were quite low, and Amy and I were talking about maybe why that is, but it may be the sort of freshwater environment there allows the plants to grow much lower in the tide frame. So how do elevation of the wetlands relate to some of the carbon metrics that we've been measuring? So these are just the marsh samples, excluding our other habitat types. So these are, each point is an individual core in the study, low marsh and high marsh pooled together. And on the left, you'll see the carbon concentration. So there's the percent of carbon in the surface samples and how that relates to tidal elevation. The vertical dashed line is mean high water, so a Z star of 1. Again, above that is typically considered high marsh, and below that might be considered low marsh. And then the right panel shows the relationship between carbon density in surface sediments and tidal elevation. So there's a lot of scatter in these data, but the data are numerous that we're able to sort of draw some patterns out here. And it's relatively clear that it's our high marshes, those that are highest in the tide frame, that are storing the majority of the carbon. These are more plots of marsh elevation in carbon metrics. In this case, it's stocks. So I calculated stocks to 30 centimeters, so Boone's top 2 layers, and then a meter, so the top 4 layers that Boone sampled. These relationships aren't quite as strong, but again, sort of a positive relationship between elevation and carbon stocks in marshes. Okay, if we look at just the tidal swamps, though, there really is no relationship here. So we do have a range of elevations in the tidal swamps, but there was no sort of clear pattern between carbon content and carbon density with tidal elevation. Yeah. I've done a lot of squinting at these data sets, and yeah, there could be, but it's yeah, it's hard to know sometimes with when the sample sizes. I mean, this is probably the best tidal swamp data set ever collected so far, but you know, you always want more data so that you can look at relationships here. But yeah, there might be some curvilinear relationship there, but nothing you could publish with just these points. Yeah. Okay, and then the last one would be the seagrass elevation and carbon metrics. So again, not a significant relationship, a lot of scattered here. But seagrasses do go into the subtidal, and all of our sampling was sort of a low intertidal. So it's possible that if you included subtidal seagrass plots as well, that elevation may play a role, particularly because the plants could get more and more light limited with depth. All right, so a summary of the wetland elevation results. Higher elevation marshes within the Pacific Northwest tended to have greater soil carbon content, density, and stocks. Elevation did seem to be less important of a driver, at least with our limited data in the seagrass meadows, and also in the tidal swamps. So salinity may actually be a more important driver of carbon stocks in tidal swamps than elevation per se. So some take-home messages from the ecosystem drivers piece of this work are that larger scale climate drivers don't necessarily seem to be strongly related or even weakly related to carbon stocks within the Pacific Northwest. But this is a question of interest to me, and I'd like to test this with sort of larger climate gradients throughout the Northeast Pacific. Site level variability in salinity may affect the carbon stocks. We've done taken a brief look at the tidal swamps and need to look at the marshes next, where we have sort of the best chance of seeing these relationships. Elevation explained little variability within the eelgrass meadows or the tidal swamps, but there was a fairly consistent relationship in the marshes. So this suggests that high marshes are areas that you may want to focus conservation efforts on, and low marshes, I think, which would be consistent with what we've heard today, these are areas that we may want to focus our restoration efforts on, where they have the capacity for higher carbon sequestration. And our next steps are to continue analysis of the groundwater time series and look at the water table effects on carbon stocks and density. And then, as I mentioned, sort of examine the climate question with a larger gradient throughout the Northeast Pacific. All right, I think it's a break now, unless you have any questions.
Just in the Northeast at the Waquoit Bay Reserve, there was a long-term project looking at, it was basically real-time flux data, so it wasn't necessarily correlated with stocks, which is what you did. And there, I don't think they looked at rainfall as a driver, but there was a modeler who basically looked at all the different variables tied to the fluxes, and by most CO2 primarily.
CO2 fluxes.
And as you would expect, the primary driver was sunlight, so it was PAR. And so I'm curious, like, maybe it's just the homogeneity of the sunlight or across the region Might be one reason why that didn't show up.
Yeah, I mean, there wasn't much difference in irradiance from, Humboldt Bay up to Puget Sound, really. I think if we included, the more Mediterranean areas of California, and, maybe went farther up into British Columbia and Alaska, for example, that we would see more gradients in irradiance that may sort of reflect that. But, plants also get light saturated really quickly, so that if you look at sort of a PI curve of a plant or an alga, they don't necessarily need full irradiance to have full photosynthetic capacity. So in retrospect, maybe it wasn't the most interesting thing to look at, but we did have this kind of cool data set that wanted to take a look at it.
Yeah, I'll go first. So I've been thinking a lot about this sediment, carbon storage and decomposition, and the question of should we expect decomposition decomposition in the sediments to be different across all of these systems? Because pretty universally, sort of the oxic reduction, nitrate reduction, sulfate reduction, methanogenesis is pretty universal around the globe. So are the stocks less a function of the conditions in the sediment and more the input and the quality of the ability or recalcitrance of the carbon that's going in there? So the decomposition rates should be pretty similar. I mean, the null hypothesis would be they're similar because the microbial communities are similar, and it's the recalcitrance of the inputs of carbon that would make those stocks be bigger or smaller. And I'm kind of thinking through this, and I don't know. So the tea bags, because they are so consistent, we might not expect those rates to vary among the different sites if the microbial communities are universal. But different types of carbon, whether it's Sitka, spruce, detritus, or might be different. So I don't know. The fact that we don't see those patterns doesn't necessarily mean that they aren't good questions and our questions aren't working, but it's...
Well, so, from my reading of the literature, and the decomposition data are like all over the place. If you ask how is decomposition related to salinity, and you find 10 papers, you get 8 answers, right? So decomposition is really controlled. I mean, it's controlled by the biochemical composition of the material, as you mentioned, but it's also controlled by the microbial community, which is almost never measured. Who's there, what organism, how abundant are they, what is the species composition of the microbes. But then the third thing is sort of the environmental context, and that could include the redox potential, include oxygen availability, sediment saturation, salinity. So it really is a tangled web. I mean, to do decomposition measurements well, I think you need to measure all of those things simultaneously, which is, beyond the scope of this work. So does that answer your question? I think that the composition is part of it, but we have some data from San Francisco Bay, for example, where we did a common garden experiment. We took 4 different plant species and put them all in the same marsh, hoping to control for environment and inundation and things. And there were some differences. Or no, actually in that data set, there were very small differences. So we had 4 common Bay Area plants, and they all decomposed at roughly similar rates over the first year. We did a companion experiment where we took one type of litter and we put that in 4 marshes that went from freshwater tidal to San Francisco Bay, which is quite saline, and we did find differences there. So that, not perfectly controlled study, but kind of suggested that salinity was a driver there.
Yeah, and I think the salinity piece is, so salinity and elevation, where we saw some possibility of trends, could be a proxy for sulfate, which is a terminal electron scepter. So if you have a lot of sulfate, then it's easier for microbes to degrade organic matter in waters that are more saline, and that's, so I don't know, it's very possible that the pattern we're seeing related to the relationship between salinity may be that sulfate reduction is helping organisms break down, the microorganisms break down carbon where it's not in the other markers.
Yeah, for that subset of microbes, but there, you have other non-sulfate using microbes that are decomposing as well.
Yeah, but they're all really So these are, you go around the globe, and they're pretty universal, and they're layered in terms of very specific redox ranges. And I mean, I think I'm less concerned about the microbial diversity question on those because they're pretty universal. But I'd like to think, I mean, as we talk about wrapping this up, I'd like to think more about what's really going on in the sediments.
Yeah, I mean, it would be great. Like I said, as I've gone through the literature on decomposition, it seems like a story that has a lot of work yet to be done and to figure out decomposition in tidal wetlands, period, yeah.
Yeah, Chris. So
Oh,
a few additional data points on this. I'll pull together, try and shorten a long story. When we worked in the Middle East, Lisa published a paper of our data set for Abu Dhabi, and soil carbon stocks were relatively low. And possibly a couple of things going on. We were trying to tease out a story about thresholds. Relatively very granular material, so high percolation rates, movement of oxygen through the sediment, at the same time low productivity. And so carbon's just not building up, soil's not building up the conditions to allow for carbon accumulation. It's getting mineralized too quickly. Coming to the US, when James Holmquist looked at the US data set, carbon density didn't vary that much around the country, except in Southern California, where, again, very sandy sediments, there's not a lot of fine material coming down, but there is some river flow, productivity, and so there seemed to be a threshold there. And when we think about just postulating here, when we think about the Pacific Northwest, all of them are higher levels of rainfall than the other places we were talking about there. But is rainfall really the parameter? Because all your systems are driven by river flow. And so you could have low rainfall in a particular location over here, where it's been raining like hell in the mountains, and you've got a lot of fresh water flowing through the system. So maybe you're not that sensitive to the local rainfall conditions. You're more sensitive to the river hydrology conditions in the Pacific Northwest.
Yeah, I think that's a great thought. So there's a great study that was done in, I think, 2009 out of EPA, and I go back to that all the time. So Henry Lee and Cheryl Brown led sort of a classification of all the Northwest estuaries, and they looked at things like the amount of fresh water that went into the watershed, and they sort of normalized the volume of the estuary to the input, and they came up with numbers. I would love to do that, but those data don't exist for all of our estuaries. So we could do that for maybe half of our sample sites, but it's just not a big enough sample size to start looking at relationships. But I think you're right, and maybe again, sort of in the beginning thought precipitation might be a proxy for that, but probably it's a lot more complex. It's sort of the amount of fresh water through the watershed.
Just to add one last comment that I forgot to mention. But also with the river flows come fine materials. Unlike Abu Dhabi and unlike places like Tijuana, high permeability because of very coarse sediments. Once you start bringing fines in, your permeability goes down. Therefore, your oxygen levels go down, so you get across the threshold. So the presence of the river, as well as maybe the influence of hydrology, maybe it's influencing sediment texture, and that's a feedback.
Yeah, and Boone's team will be able to look at that. So excited to see those results. Yeah.
Just a quick question.
Oh,
Did
okay.
you analyze low marshes and high marshes separately relative to elevation?
I didn't, but we could. Yeah. Yeah, we could look at that. So. Question back here. Sorry.
Yeah. Well, actually, that whole conversation gave me a thought. Have you thought about crossing the NHD watershed boundaries with your PRISM data, which should give you a pseudo amount of water that's coming down that system?
Yeah.
It'd be rough, but it'd give you something to compare.
Yeah. I mean, short answer is no, but thank you. Good idea.
And then my other question was, did you say you measured pH out there? Or is that something
Boone's
that-
team measured pH, sort of
okay.
spot measurements, yeah.
Okay. Did you look at anything relating to the pH and any of these other, and the carbon parameters? And
No, I haven't
decomposition?
taken a look at that.
Okay, I was just curious.
Yeah.
We have throughout the world found that we don't see any real correlation. And the reason why we really do pH is if we know it's below 7, because there's very little inorganic carbon, so we don't have to do the additional work. So what we're really trying to do is just to make sure that we
That was it.
All right, okay. I think it's break time. You get to hear me again, so if you have any more questions.
Okay, let's take about a 10-minute break.